Hello, everyone. Please accept my greetings from Mayo Clinic, Jacksonville, Florida. Uh, I personally thank the organizing committee of Australian Space Biology Symposium for inviting me, to deliver a talk and to share my research to a greater audience, great researchers. So today I'm going to talk about one of my research strategies to protect, means technically to mitigate the GI tract from radiation injury. So before I move forward, let me take the opportunity to introduce myself in brief. I am basically a radiation biologist. I did my PhD at Institute of Nuclear Medicine and Allied Sciences. Means and then I moved to get my postdoctoral degree at Armed Forces Radiobiology Research Institute, which is a kind of flagship organization, is doing all sorts of radiation countermeasure development research. And Armed Force Radiobiology Research Institute is part of Department of Defense, United States. And after my first postdoctoral experience, I moved to MD Anderson Cancer Center as a junior faculty, and recently I moved to Mayo Clinic as an assistant professor in the radiation biology department. So now let us talk science. So let me share my screen with you. So I know we have lots of eminent speakers to deliver a talk on space biology field, but still I would like to start with the basic problem. When we say protecting or mitigating GI tract from radiation injuries, we mean radiation exposure which causes acute radiation syndrome, means ARS. And what is ARS? ARS is when we receive radiation exposure, means high dose of radiation exposure for a very short time period. And based on the exposure we receive, ARS is categorized into three distinguishing type. One is hematopoietic syndrome, which causes hematopoietic syndrome, and another is gastrointestinal syndrome, and the third one is cerebrovascular syndrome. My research in this talk would be focused on GI syndrome, means the radiation that causes GI syndrome. So let us talk about the potential sources that can cause ARS. So we all know that we are having severe threat of getting exposed to radiation. So the main source of radiation exposure could be the nuclear bomb. That is the extreme phenomenon, I would say. Improvised nuclear device, means a dirty bomb, radiological devices, and natural or man-made accident of nuclear power plant. So you can see this is a really old map. Means we have lots of nuclear power plant all around the world. If I include the nuclear devices, means which we can say that radiotherapy equipment used in clinic, this map turns almost red. Means everywhere we have a threat to get exposed with high dose of radiation. So let us talk about the possible countermeasures to ionizing radiation. So there are main three types of radiation countermeasures. One can think one to prevent the initial radiation injury, another to repair the molecular damage caused by radiation, and the third one, and which is the most recent one are those means those agents which stimulate the proliferation of surviving stem and progenitor cells. So these are three main types, and you can see the different category falling in these three types of radiation countermeasure. So when I say radiation countermeasure, it means it has two segments. One is called protector and another is called mitigators. So protectors are those which are given prior to radiation exposure. Means if we receive drugs before the radiation exposure, and when means agents are given after the radiation exposure, we call them mitigators. Technically, we need mitigators, not the protector, because we don't know when we can get exposed to radiation. So technically, we need mitigators, not the protectors. But in loose language, people combine these two terms and say the protector. But technically, if we define the combination of protector as well as mitigator falls in radiation countermeasure category. So let us talk about, as I mentioned, that this talk will be centered towards the GI syndrome. So let us talk about the intestinal crypt rescue. So the role of R-spondin has been shown to protect GI tract from radiation injuries. The simple logic behind the use of R-spondin is to enhance the number of intestinal crypts, which harbor the intestinal stem cells, obviously. So the better the number of crypts we will have the better chance of survival. So here you can see if you have 100 crypts, suppose we have 100 crypts and we get the radiation exposure and only 5 to 10% crypts survived, that may lead to death. But if we double the number of crypt and still we are getting the same radiation exposure, we will have higher number of crypts, although it is again 5 to 10% surviving crypt, but we will have better chance of survival. This is the logic. But there is the limitation of using R-spondin. Means we can categorize it in two ways: limitation related to activation of Wnt signaling and limitation related to physical or physiological barrier. So means you can see that number of cofactors could be a limiting factor. So when R-spondin, if we use R-spondin, it activates LGR4, 5, 6, and then the HSPZ, and then it finally gets into the Wnt-catenin pathway. So there are multiple cofactors. If one cofactor is not able to respond, then the ultimate Wnt-catenin pathway won't be active. So there is the limitation at the level of cofactors. There is the limitation at the number of receptors. So we need the proper receptors. So if there is one cofactor which is not acting properly, the efficacy would reduce by 25% if there are 3 or more cofactors involved. And then we will talk about the physical or physiological limitations later on in next slide. So let us talk about the structure of villus. You will notice that there are two pathways who act coherently for the development of complete villus. So when Wnt is on, it helps in proliferation of crypt cells, but we need Notch signaling to help the crypt differentiate into the villus, complete villus structure. So, so Wnt on means crypt proliferation, but we need not on off and off mechanism to differentiate the crypt cells. So not off leads towards the differentiation of enteroendocrine cells, and not on leads to the differentiation of absorptive cells. We postulated that differentiation as well as means proliferation as well as differentiation of these crypt cells is really crucial for protecting, means mitigating the effect of radiation. So here you can see we have healthy crypt and villus, and we get the ionizing radiation. So villus is shattered, villus lining is shattered, and so we have a endotoxemia, means bacterium, bacteremia, insufficient absorptions, and loss of fluid. So no crypt proliferation and no crypt differentiation, death of the organism. But if we have crypt proliferation but no crypt differentiation, means our GI tract may not be healthy, means considered healthy. So here is the catch. If we use something that activates Wnt and something that activates Notch, could be better than one drug alone. So we used recombinant Wnt3, means this protein means why to use something else to activate Wnt if we can use Wnt itself? That was the logic. And valproic acid to activate the Notch signaling. So here is my experimental design. First, we tested in vitro. So means we use the organoid intestinal crypt method. So we isolated crypt and cultured in 24 well plate, and on day 3 we exposed all these crypts with 2 gray of radiation, and 24 hours later all these crypts received recombinant Wnt3, means 50 and 100 nanomolar, and valproic acid 1.5 millimolar. So, and on day 10, we scored all these crypts. So we found and you can see clearly that combination drug had higher surviving crypt in comparison to means radiation alone group, and in comparison to single drugs, and you can see here that the burden per organoid was also significantly higher in the combination treatment group. So this gave us the idea that the combination drug could help in mitigation of radiation. So we later did the immunohistochemistry in vivo, means mice model. So we irradiated 10 gray total body radiation, and 24 hours later, means we gave this combination drug 100 microgram per kg body weight, and valproic acid 300 milligram per kg body weight. And on day 4, these mice received fitsidextrin, because we wanted to see the permeability of means intestinal permeability of these mice. And on day 8, 1 hour before the euthanasia, we gave BRDU. We wanted to see the proliferation, means efficacy of these crypts in intestinal crypts. So I would like to mention here that if someone wants to test radiation-induced damage, the 4th day or 3.5 day is the crucial, means one should harvest the intestine on day 4. But if someone wants to evaluate the recovery, means the recovery means proliferation, then it would be day 8. So we picked both days, 4 day and 8 day, for 2 different purposes. So the first thing we tested that uh, does this Wnt3 means enhance the beta catenin expression in Crypt. So we found that the combination drug has higher means expression of beta catenin in Crypt. So and we tested it on day 4. So it gave us the idea that our combination drug is able to 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 act on the Crypt and enhance the effect of means enhance the protective effect by enhancing wind signaling. So then we analyzed on day 4, as I explained, and day 8, the overall health of intestinal lining. So we evaluated the mean Crypt number, villus length, mean mitotic figure, and you can clearly see that combination drug, valproic acid and Wnt3, means increase the number of Crypt, means and as well as the length of villus was also higher. So you can also notice here that on day 4 it is higher, but on day 8 the length becomes even more longer. So means it shows it gives us the preliminary idea that this combination drug is able to give protection to the GI health, means against the radiation exposure. So this is the representative figure. You can see here that day 4, the villus is really a stunt, and there are very less number of Crypt here, and here it is the day 4 and day 8. The villus length is really means longer, and we have large number of Crypts here. So as I explained, on day 4, we evaluated the intestinal permeability, and on day 4 as well as day 8, we evaluated intestinal permeability as well as anti-inflammatory response. And you can see that serum dextran leaked into the blood, means we gave it through the oral gavage, and it leaked into the blood in the radiation alone treatment group, means and here this PBS means that this receives the 10 gray of radiation and receive the PBS only, and you can see that combination drug had less blood serum dextran, means and here you can see the alone Wnt3 and valproic acid, although they did a little bit better, but not much. So combination drug is quite better than alone and with the means just the radiation treatment group. And we here this data also correlates with the anti-inflammatory response, where you can see that the combination drug has less means inflammatory response in comparison to the radiation itself. Then we evaluated the effect of these drugs, these combination drugs, on DNA damage, double-stranded break. So we evaluated this by apoptosis, means tunnel assay, which means evaluates the DNA damage. And you can see here the combination drug means had less number of DNA damage in comparison to the previous treatment group. And on day 8, it further recovered. So means all these machinery helped them to recover from the radiation induced damage. So here you can see that we further evaluated the effect of all these two combination drug on radiation induced DNA, means apoptosis. So we evaluated the cleaved caspase 3, and here we have two different graphs. One tells the cleaved caspase positive cells per crypt and cleaved caspase positive cells per high power field. And in both way, we found that Wnt3 and valproic acid combo drug has these treatment group has less number of cleaved caspase, which correlates with the apoptosis. So it protects it is able to protect the GI from the radiation induced damage. So we further wanted to know, so what about proliferation? So does it proliferate? So we saw that on day 8, means I mentioned that the proliferation one should test on day 8 after radiation. So we tested it on day 8. So, and we found that that combination drug has a higher number of BRDU positive cells per crypt in comparison to radiation, it means irradiated group, but receiving just PBS. So, and then we wanted to test the PCNA, which is a kind of proliferation marker. And we found that on day 4 as well as day 8, our result correlates. So further, we wanted to evaluate the differentiation. So, okay, we found cell is able to proliferate and cells is able to protect from the DNA damage and stop the apoptosis, but does it really help in differentiation? So we found that the number of goblet cell per villus was quite higher in combination treatment group in comparison to the irradiated and PBS receiving group, as well as the number of Paneth cells per crypt was also higher in combination treatment group. And here you can see that number of enterokine also higher in combination treatment group. And I apologize, the graph has a little bit shifted. So what you can see that here, that means there are lots of goblet cells you can see in the villus, and here it is quite less. And this is the unirradiated treatment group. So it almost looks like that it looks like a normal unirradiated group. But however, you will see in later slides, so this something wrong happened, means it didn't get translated into the survival. So we have survival data later on. I'll come on that in a couple of minutes. So further, when we had the evidence that this combination drug is able to mitigate the effect of radiation on GI tract, so we designed the experiment where we gave total body radiation of 10 gray, and then 24 hours later, we gave the combination drug, recombinant Wnt3 and valproic acid, and did the survival study. So we planned another study again. So we wanted to, this time we wanted to enhance the dose. Means we gave 10.5 gray and 5 gray of radiation to the GI tract. So we wanted to see that if we irradiate just GI, what happens, means not the total body. Although in real scenario, we will receive the total body radiation, not the GI only. But this means this design was to know that if we have just the GI protection, then it can be correlated in a clinic, means where people receive the rectal radiation or GI radiation for different kind of cancers. So here is the survival graph, and you can see here that the graph has shifted considerably, and we had 20% survival. And and one can say that, oh, yes, you have 20%. This is a lot, because once you give radiation, with total body radiation, your means the mice means animals body is shattered, and I'm not giving drug just immediately. I am giving drugs 24 hours later. So this is a really good data, and we wanted to know what happens if we irradiate just the intestine, not the total body. And here you can see the partial body radiation, means I irradiated just the GI, and you can see that we had 100% survival. Means mice received a total of 15.5 gray of radiation, and that is really significantly, means considerably very high dose of radiation. So we had total 100% survival. So the other experiment we did, that what if we just sealed one leg, means to one femur, to have at least means minimal homeopathic spillage. So means uh, so far you might have understood here that mice died just not because GI syndrome, because when we expose mice with a total body, their homeopathic system is also shattered. So it is the combination of GI as well as homeopathic syndrome. So if I am protecting GI, mice still die because of the homeopathic syndrome, and our system is just protecting the GI. So in the another graph, this graph, when we irradiated just the GI, we had 100% success rate. So this is the really wonderful thing. So in summary, we can see that means proliferation as well as differentiation of crypt cells is required for mitigation, and means all these combination drug means reduce the inflammation of intestinal permeability. It inhibits the apoptotic pathway, and it helps in cellular proliferation. And so that we can say that it could be developed as a novel radiation mitigator. So this is our lab, and I work with Dr. Sunil Krishnan, and he has really done a great mentoring to me, and we work with him still at Mayo Clinic. Thank you.